Okay, so this is starting part 1 of the blood. This video will not have all of it. My voice is already struggling a little bit, so I probably will have to wait and do video 2 of part 1 for the blood tomorrow. Okay, so first of all, just a few basics. Now, the order in your book, they cover the heart first and the blood vessels. We do it differently. Basically, we're going to do it in the same order as we do lab, so we're actually going to cover blood first. Okay, but the heart, blood vessels, and blood, of course, are all part of the cardiovascular system. So the heart, of course, is the pump. It's what keeps blood moving. The blood vessels are the conduits or pathways that allow blood to get to everywhere in the body. So the blood, depending on the person, is about 5 liters of blood. Females 4 to 5, males 5 to 6. It is a connective tissue. It's one of the few types of fluid connective tissues we have. The other one is lymph fluid. It makes up about 8% of the total weight of your body, and of course is always moving, always circulating through blood vessels in your body. So when you take whole blood and you let it sit for a period of time or centrifuge it, it's going to separate out into 3 different components. 2 are major. So one of the first major is the one here that is the most superior, which means it's the lightest. That's going to be plasma, which of course the main component is water. And it is an extracellular matrix of the blood. Okay, so inside the blood we have cells, and it's the fluid that's surrounding all of those cells. Serum is what happens after blood clots. So it's basically the plasma minus the fibrinogen, which is one of the plasma proteins and other clotting factors that are made by platelets. You cannot see that here. Basically, when they take a tube of vial blood, they're actually going to have an anticoagulant in it, so it will not coagulate, and they can do the blood test that they need to do. The other 2 components that can be seen here include the formed elements. So the formed elements is the rest, which is roughly 45%. 44 of that is by your red blood cells or erythrocytes, and they are heavier than what makes the buffy coat, which is about 1%, and that's going to be the rest of the formed elements, which are going to be the white blood cells and platelets. Okay, so the major 3 types of formed elements. Erythrocytes and leukocytes are cells. Platelets are not. Okay, so again, that's why we use the term formed elements instead of just cells in blood. So erythrocytes, of course, are also called red blood cells. Leukocytes are also called white blood cells. And then platelets are the ones that are actually fragments of cells. You can also call platelets thrombocytes. Okay, so the different layers we're talking about, I already mentioned this. Okay, so what is blood important for? Basically, what does it do to help keep our body in homeostasis? First of all, blood is the medium in which gases can be exchanged. So we're going to be able to move oxygen into the cells so they can make ATP to carry out the activities. And as cells are undergoing cellular respiration and making ATP, they give off CO2 as a waste gas. So we have to remove that from the blood. It allows us to distribute different solutes. A lot of these are going to be electrolytes or ions. Can also be nutrients, glucose, lipoproteins, amino acids, hormones, other metabolic waste. It does have immune functions. So because one of the formed elements in blood are leukocytes or white blood cells, they play a major role in protecting the body. There are also proteins that are in plasma, and these proteins can bind to bacteria and inhibit growth. Of course, blood also helps us to stabilize blood pressure. It is the major factor that determines blood pressure. The heart, obviously, how it contracts is the other. Blood also helps to help us maintain body temperature at 37 degrees Celsius. So it will carry away heat, basically bringing it closer to the surface to dissipate heat. When you're cold, we will hold blood closer to the core, helping us keep warm. So why is blood slightly warmer? That's because of all the chemical reactions that are occurring in it. In the body, we know that heat is given off as a byproduct when you convert one form to the other. And so that heat is transferred to the blood. Blood also functions in blood clotting. We have platelets and certain proteins that will form a clot that essentially, it's a clump of tissue that will stop the leakage or bleeding of blood until the tissue can be reformed. And it also helps us maintain our acid-base balance in the blood. If we can maintain pH balance in the blood, then essentially the pH of all the other body fluids should be able to be maintained. So you need to know the range, 7.35 to 7.45. And we can keep it in this very narrow range because there are several buffering systems that help us stay there. Remember, a buffer is going to be a weak acid or base, one that doesn't fully dissociate. So it helps resist changes in pH. We discuss a lot more acid-base balance and buffering in the last chapter for this semester. Plasma typically has a yellow tint to it. It's over 90% water, depending on the book, 90 to 92%. It is one of the factors that determines the thickness. So blood is thick, but it is still a fluid. And of course, that's because of the plasma. The less water that's in the plasma, of course, the thicker the blood. And it means that blood will move slower and more sluggish, which is not good. Okay, so it's important that you have at least, or that you have close to 90% plasma in the blood. Plasma proteins form, we call it colloid, and they make up about 9% of the plasma volume. The several different plasma proteins we have, albumin definitely makes up the majority. We have immunoglobulins or immune proteins, which we also call gamma globulins, transport proteins like protein carriers or lipid carriers, and clotting proteins that, of course, are going to be part of stopping bleeding. Okay, so if we talk about what are the differences or functions of these plasma proteins, as I mentioned, albumin is by far the one that we have the most of. It makes up over 50%. It's a very large protein, and like most plasma proteins, it is made by the liver. It's responsible for maintaining blood's colloid osmotic pressure. What does that mean? That basically means plasma proteins, and albumin's the largest one, it constantly pulls water towards it. So it ensures that some fluid is remaining in the blood, even though you have constantly fluid moving out towards the cells. Again, it's one of the main proteins that also regulates the viscosity of blood, is responsible for that. Immune proteins are gamma globulins. We call these antibodies, which we talked about with blood. They're made by leukocytes, and they are a component of the immune system. We will talk a lot more about antibodies when we do the immune system. Other plasma proteins, transport proteins, so that lipid-based molecules that are hydrophobic can travel through the water. Clotting proteins, again, to stop the bleeding, and these are going to, the clotting proteins in blood, basically when you're bleeding, become active, and those along with platelets help stop the bleeding. So we're going to discuss that process in much more detail later. So that makes 99%. The last 1% are very, very small molecules that are dissolved within the water portion, forming a solution. And we've actually talked about several of these small molecules before. So this would include glucose, any of the amino acids, ions like sodium, chloride, potassium, dissolved gases, oxygen, and carbon dioxide, as well as smaller metabolic waste. Okay, so here's a chart that basically breaks down the different components of plasma. Okay, as I mentioned before, the liver produces a lot or most of the plasma proteins. When you have liver disease, which we call cirrhosis, that can actually be caused by several different things. It could be cancer of the liver, from alcoholism, long-term use, or from hepatitis, viral hepatitis. It is somewhat common in the U.S. It leads to more deaths in men than women. Typically, this is because of the alcoholism. So because the liver makes plasma proteins, as the liver becomes stiff and hard and basically is unable to do its job, that's what cirrhosis is, it cannot make the plasma proteins. So you decrease the colloid osmotic pressure, so you don't have as much fluid in the extracellular spaces, which makes you basically start to collect fluid in cavities outside of the body. Having edema in the abdomen, it's called ascites, and basically it almost looks like a person will be pregnant and it's just fluid. And typically at that point they'll have to have the fluid drained, depending on how much they're producing, 2 to 4 times a week. They have a decline in the clotting factor, which of course means that they can't clot as much. So they're going to bruise very easily, and it's going to take them much longer to stop bleeding when bleeding does occur. Okay, so some review. Blood plasma and interstitial fluid are both components of extracellular fluid. That is very true. Okay, which of the following is not true concerning blood and how it transports? Nutrients like glucose and amino acids are carried from the gastrointestinal tract into the blood so it can go to the rest of the body, so that's true. Carries oxygen from the cells to the lungs, that is the opposite. We want oxygen to go from the lungs to the cells, so of course that's the one that's false. Blood also gives an opportunity for other chemical messengers like hormones to travel and to get to where they need to go. It is a way for us to carry waste products for elimination, not only CO2 gas that will be dropped off in the lungs, but also metabolic waste that we mainly will dispose of through the kidneys. Okay, what is the volume of blood in an average adult male? Since males are typically larger, it's going to be about 5 to 6 liters. Which statement about blood is not true? We already mentioned this in one of the questions. Blood plasma is an extracellular matrix. 90% is water. Most of the proteins are made by the liver, and 55% of the blood is made of plasma. So here it says about 10% of the formed elements are red blood cells. Clearly that's the one that's false because the others are true. That should be about 44%. And which one of the following is not a formed element found in blood? Those all are. White blood cell includes 5 different types of cells, one of those being lymphocytes. Red blood cells, and then of course the only type of formed element that's not a complete cell, which is the platelet. Okay, now we're going to talk about erythrocytes and how they carry oxygen. So the shape of a red blood cell is designed to be able to carry oxygen adequately and be able to move through the tiny spaces of a lot of the capillary beds. So a healthy red blood cell will have this biconcave disc that is thinner in both sides, so it's concave on both sides, the edges being thicker. Having this increases the surface area, which means it can carry more oxygen and CO2 to where it needs to go. It also ensures, like we're seeing here in this picture, very, very small capillaries, you have one red blood cell at a time. So it gives them some flexibility to move through capillaries, which are not straight. Sorry, I'm starting to lose my voice a little bit. Mature red blood cells do not have a nucleus. That's what a nucleate means. Part of the maturation process, they just lose it. It also does not have most other cellular organelles. The advantage to that, mitochondria take a lot of ATP. We don't have to repair red blood cells. We just get rid of them when they're old and make new ones. So because you don't have this cell that's filled with all these organelles, you have a lot more enzymes. And the main component of red blood cells is hemoglobin, about 95%, and that's all proteins. And that's a big component of how the red blood cell is flexible as well as the shape. So these both allow for stacking of the cells, which again ensures that you won't have a blood clot and possibly block or occlude blood flow further down. It's called the rouleau formation when they stack like that. Okay, so you can see superior view. Of course, we have the concave center. From a side view, you can clearly see it's thinner in the center, which is why when you look at it on slides in a light microscope, the center might even look white or very, very faint or pale. And this is just showing how chocked full it is of different enzymes and hemoglobin. This discussion question, we just kind of talked about it. Not having a nucleus or other organelles doesn't require as much energy, and it allows for space for those proteins, hemoglobin, which again are important in allowing red blood cells to move throughout capillary beds and other vessels. Okay, so again, the main component of red blood cells is hemoglobin. Hemoglobin is a very large protein. It's what we call a quaternary structure of a protein because it has 4 different polypeptide chains, so 4 different proteins essentially that make it up. You have 2 alpha chains and 2 beta chains. Each polypeptide has an iron compound. In the center of the iron compound, we call that a heme group. And what's important about the heme is that that is what binds to oxygen when it's oxidized. The more oxygen that is being able to be bound to the heme, the brighter the molecule, the redder your blood. So redness of blood comes from oxygen, and that again is because of iron. So it's going to be oxygen rich in the lungs, and then when it gets to the cells, the capillary beds in the cells, it's going to drop off the oxygen and pick up the CO2. And that's when the blood becomes darker in veins. So we call it oxyhemoglobin when oxygen is bound to the hemoglobin. And once it's dropped off the hemoglobin, deoxyhemoglobin. Is blood forever blue? The question is no, it's not. Okay, so this is just roughly showing the 4 different polypeptide chains. Again, the most important of that is the heme group. The very core of the heme is going to be your iron, and that's where we're going to bind the oxygen. Well, not only does oxygen bind to hemoglobin, carbon dioxide does as well. So when carbon dioxide is bound to hemoglobin, we give it a different name, carbaminohemoglobin. And basically the less oxygen that's present, the more carbon dioxide is going to bind to the hemoglobin. About 23% of the amount of CO2 that we're transporting in the blood away from the cells is carried bound to hemoglobin. It doesn't have to bind to the iron heme, it's just carried by the red blood cell itself. Hemoglobin also binds to carbon monoxide to form carboxyhemoglobin, which binds to the iron core. In fact, it binds stronger than oxygen does, which is a good thing. So at these cells, capillary beds, we can pick up CO2, which wants to be bound to the hemoglobin. So as it's wanting to bind hemoglobin to the carbon dioxide, it's going to be dropping off the oxygen at the cells. This does change the shape of the hemoglobin, which unfortunately means that the red blood cell cannot drop off oxygen. So cells become starved of oxygen, and if that occurs for too long, they will die. Looking at these, I really don't expect you to know the chemical equation or anything like this in here. Really, all I want you to see is, again, that the core is iron, and oxyhemoglobin, notice that oxygen is bound. Deoxy is when oxygen has been unbound and is taken to the cells. Okay, erythrocyte structure. This is actually a real picture of hemoglobin. It's highly magnified and using a colors to it. It's a type of scanning micrograph. So you can see, I'm not sure what colors indicate what, but obviously 2 of these are going to be your alpha polypeptides. The other 2 are going to be the beta. And again, it makes that very large protein quaternary structure. So erythrocytes do not live for huge, long amounts of time because it's relatively short, again, because they don't have a nucleus in so many of the other organelles. So that's about 100 to 120 days. So basically, when they die, it's because damage has been done to them. Harsh environment, you have to think about as they go through all of those tiny little capillaries, they're banging into the walls, and they have all these things that are binding and unbinding, and eventually you basically have gunk, stickiness that appears on the red blood cells. And again, they can't repair because they don't have the organelles. So our body is continuously phagocytizing, getting rid of the old damaged ones and making new red blood cells. And those, of course, we already mentioned this, are made by the bone marrow in response to EPO. We talked about that in the endocrine lecture. You have 2 different terms here. A lot of students easily get these confused. Hemato refers to blood. A hematologist, okay, is someone who looks at blood. So hematopoiesis, poiesis is the process of. That is making red blood cells. And red blood cells come from hematopoietic stem cells. Essentially, all blood cells come from hematopoietic stem cells. You have several different forms of hematopoiesis. We also sometimes call this hemopoiesis, same thing. So specifically, when it's just red blood cells, we can then give it a more specific name, which is erythropoiesis. So we're making these stem cells differentiate into erythrocytes versus any of the other cells in blood. Erythropoiesis takes about a week to complete. So the stem cells basically will start to differentiate, start to mature. They have progenitor, then we call it the erythrocyte colony forming units. And again, these, once they become CFUs, they have to become erythrocytes. If it's still a progenitor, that is still a form of a stem cell. So these CFUs then will become immature red blood cells, which we call pro-erythroblast, when EPO is released. So you can have a lot of pro-erythroblast in the bone marrow kind of waiting to get to that final maturity state. So when EPO is released, it will stimulate them to quickly to basically further the maturing process, become erythroblast and then the erythrocyte. The EPO, again, is made by the kidneys in response to hypoxia. Hypoxia exists when oxygen levels are low. So when oxygen is low, your cells are not getting the proper oxygen. So the kidneys sense that there's not enough oxygen, so they make EPO, which makes the bone marrow make more red blood cells, and red blood cells carry oxygen to cells. basically your kidneys don't necessarily know that you might not just have oxygen because it's a high altitude. So this is just going through the different steps, and this is really why I wanted this picture up here, was because this is showing where this is going on. Remember, bone marrow is found within spongy bone, which essentially is in several different bones of the body. In the adult, it's in the proximal epiphysis of the femur and the humerus, as well as in all flat bones. Okay, so again, you have the stem cell, then it goes to the erythrocyte, CFU, the proerythroblast. The proerythroblast, again, we just take off that becomes the erythroblast. That very, very quickly starts to make hemoglobin. And basically, the more hemoglobin and other proteins and enzymes that are added, the nucleus starts to get smaller and smaller, and the other organelles are removed. Eventually, we basically push out the nucleus, and then that becomes our reticulocyte, which is also just referred to as an immature red blood cell. These can enter the bloodstream. So technically, they are not mature. And in fact, if you have a large amount of these in the bloodstream, that can actually indicate that there's some issue with your bone marrow being able to make adequate red blood cells quickly enough. Okay, so early erythroblast, late, basically the late just pushes out the nucleus. As soon as the nucleus has been pushed out, then it's the reticulocyte. It's going to continue to get rid of all the extra stuff that it doesn't need, continue to be filled with hemoglobin. Again, this can enter the bloodstream reticulocyte, or they can stay in the bone marrow, continue to mature, and become the full red blood cell. So we mentioned this before, just to go over it again. Let me use the picture. Erythropoiesis. So the kidneys are triggered to release EPO when blood oxygen is low. Okay, so this is your set point. It goes too low, so we have to do something to raise it. So of course that means this is a type of negative feedback, like most things are. So the kidneys detect the low oxygen. They make EPO. EPO immediately enters the blood vessels in the kidneys, then makes its way to the bone marrow, where that's going to stimulate and increase how fast red blood cells are maturing. The more red blood cells you have, the thicker the blood. And the term hematocrit actually refers to the amount of erythrocytes in the blood. So the more EPO, the higher the hematocrit, meaning the more red blood cells and the more oxygen carrying, potentially, cells. So when they get old and damaged, how do they die? Erythrocytes become trapped in basically spaces in the spleen. So the spleen is the major organ where we destroy red blood cells. However, macrophages anywhere in the body can engulf a old damaged red blood cell. We have specific macrophages that stay in the spleen, so those will digest the erythrocytes. Again, the major component of the erythrocyte is the hemoglobin. So we're going to break down the hemoglobin into amino acids, iron, ions, and heme. Okay, we don't want to waste anything. So we're basically going to try to recycle a lot. So heme is broken down further. It's converted to biliverdin, which is a greenish pigment. And then later through the maturation process becomes a yellow pigment called bilirubin. Bilirubin is actually made, kept in the liver, stored, and that's going to be a component of bile. When you have too much bilirubin built up, it causes jaundice. So jaundice is a yellowing of the skin, and especially the sclera of the eyes are the most obvious. iron and amino acids, we're going to recycle and use these. So we're basically going to rebuild the hemoglobin with the remaining iron and amino acid components. So we have to take the iron back to the red bone marrow where red blood cells are formed. And it does this by being attached to a protein called transferrin because it transports the iron, mainly here from the spleen, back to the bone marrow. Bilirubin is sent to the liver, and we're going to excrete part of it as a waste product, and again, part of it's going to be used later. Okay, so this is just showing the different steps if you're a more visual person. Jaundice, which you can see again, yellowing of the skin and of the eyes, can occur by a few different ways. If the bile ducts are blocked, so basically the liver makes bile and it takes it to the small intestines, and it's also stored and concentrated in the gallbladder. If those are blocked, the bilirubin is basically going to continue to rise, which leads to destruction of tissue. Also, if the liver can't absorb, process, or excrete the bilirubin by making bile, okay, this can occur as well. After red blood cells die, how are the components of hemoglobin recycled or excreted? So remember, the heme is going to get broken into the biliverdin, which eventually becomes the bilirubin, which is going to go to the liver and become waste products and part of bile. Also, you have the iron components, which are going to be recycled, along with making up the hemoglobin. So the globin portion, those are your proteins. Proteins are made of building blocks amino acids. So amino acids like iron will be recycled. So again, jaundice occurs when bilirubin builds up and cannot be excreted by the body. Okay, so we're going to talk about basically abnormalities if you have too many erythrocytes or too few. Too many erythrocytes is termed polycythemia. Poly, too many, cy, cells, in this case red blood cells. It can be caused by several different things. So again, hypoxia, when you don't have as much oxygen in the air, which naturally occurs in high altitudes, which again is why a lot of professional athletes will train in high altitude areas. Also giving shots of EPO, which is what we call blood doping, or even removing blood from one's body, storing it appropriately, and then transfusing it right back. That's blood doping as well. Smoking. Smoking decreases the amount of oxygen that is given to the cells, so your body naturally is going to make more red blood cells. Or COPD, which is chronic obstructive pulmonary disorders, so that is hindering the ability of oxygen to get to the lungs, and therefore is not going to be able to be carried by the red blood cells as much. If you're dehydrated, this will be very temporary. Dehydration means that you have a drop in blood volume, which means you're going to have a drop in blood pressure. If we increase the thickness of blood, that's going to add a little bit to the volume, but it actually adds a lot more to the pressure because it becomes much thicker. Or it can actually be a disease, not necessarily caused by any of those things, and we give it a more specific name, polycythemia rubra vera. So when you do not have, essentially it's not exactly enough red blood cells, but that's basically the effects of it. Technically, we define anemia as decreased oxygen carrying capacity. What carries oxygen? Red blood cells. How do red blood cells carry oxygen? Through hemoglobin. So three primary causes are that you don't have enough hemoglobin. So if you don't have enough hemoglobin, you don't have enough heme, which is actually the iron where oxygen binds to. You actually have a drop in red blood cells, which we refer to that as hematocrit. Or the hemoglobin is not made properly and therefore cannot bind as many oxygen molecules to it. There's several conditions that can occur that are similar to anemia. A lot of the same symptoms, basically, that you have your oxygen starving your cells, and eventually you can have cellular cell death, fatigue. The same kind of symptoms will be there. Okay, so general symptoms include pale skin. Okay, you can actually, if you remember from pigments of blood or skin, it was made by melanin, keratin, and the third was hemoglobin. So it's that reddish pink kind of tone to your skin. If you don't have as many red blood cells, it's going to be pale. It's going to make you really tired because your cells don't have as much oxygen. Therefore, they can't make as much ATP, so they can't be as active. It's going to make you weaker. You're going to have a shortness of breath. You're going to basically try to increase breathing to try to bring more oxygen in. But the problem is you don't have a problem with bringing oxygen in. You have a problem with getting oxygen to cells that need it. Several different types of anemia cause elevated numbers of reticulocytes. I mentioned this earlier. That's your immature red blood cell. So your body knows that it doesn't have enough red blood cells, so basically it starts to release them early. Several types of anemia can even elevate heart rate, and that's basically the body's ability. The cells are still deprived of oxygen, and so they're thinking, if I can increase the pump, then I'm going to get more blood and more oxygen. Okay, so one of the examples of anemia, when it's specifically a decreased amount of hemoglobin, and it is the most common type, is iron deficiency anemia. So a person doesn't eat enough iron, get enough iron in their diet, or they cannot properly absorb it, or they're losing blood and therefore losing iron. Remember, we recycle the iron. So you will start to see the deficiency there. Without the iron, you can't make the heme group. Therefore, you cannot bind oxygen. And erythroblasts actually can't even make any more hemoglobin to make new red blood cells. 20% of all women of childbearing age have iron deficiency anemia. Definitely, and not necessarily all the time, but while they're menstruating and typically afterwards, they will have the symptoms of that. Men, it's much more rare. Another form of anemia is when it comes from a chronic disease. So you could have cancer long term. Cancer, basically, you have abnormal division of cells. So if you have a lot of activity in one area, it's going to decrease the activities in other places. And a lot of disease states will interfere with being able to carry iron from the liver back to the bone marrow. And again, if you don't have the iron, then you can't bind the oxygen. Other important factors that can cause anemia: if you are deficient in B6, that's one of the components for making hemoglobin. If you're not eating properly, of course, you won't have enough iron. Poisoning with heavy metals, one of the main ones are lead. Lead affects the proteins that are necessary to actually make the polypeptide units of the hemoglobin. And pregnancy also decreases hemoglobin levels because basically you're making a lot of the hemoglobin, the red blood cells doing nutrients for the baby. Decreased hematocrit. Again, hematocrit, we're talking specifically about the number of erythrocytes in the blood. So anemia, if you don't have enough hemoglobin, you're not going to be able to make as many red blood cells, so your hematocrit will be lowered. Of course, again, if you're bleeding, whether it's hemorrhaging from an injury, if you're just not clotting, it's just a wound that's gaping, that's leaking. or even stomach ulcers. Any of that blood loss, you are unable to recycle the iron. And so you're not going to be able to make the hemoglobin and therefore bind oxygen. Pernicious anemia is because of a deficiency in B12. B12 is a B vitamin that is necessary for DNA synthesis, which of course if you're making new cells or hemoglobin you would have to do. And one of the major causes of this is because cells in the stomach are unable to produce what we call intrinsic factor, which allows us to absorb B12. If you can't absorb B12 naturally that way, you can get B12 injections that will help reduce the severity and the symptoms of pernicious anemia. The decreased hematocrit, of course, you don't have, again, as many red blood cells. So another effect of that could be that something is killing the red blood cells. A bacterial or parasitic infection like malaria works this way. Diseases of the immune system, sometimes the immune system will trigger its own cells, and that might include, or proteins, including those involved with hemoglobin. Anything that affects the liver, because again, the liver is going to convert iron and allow it to be transported back to the bone marrow so we can make hemoglobin. Again, lead poisoning, we call all of that hemolytic anemia if it's a cause by basically an outside agent. Medications, exposure to radiation can cause what we call aplastic anemia. Aplastic anemia can actually be a lot more harmful than the others because it actually affects all hematopoietic stem cells in the bone marrow. Not only does it affect erythrocytes, but it can affect leukocytes, your white blood cells as well. Abnormal hemoglobin. The most common thing that would cause hemoglobin to be abnormal is an actually hereditary disease, which we call sickle cell anemia or sickle cell disease. And again, when you're talking about anything that you inherit from parents, you get one allele from each. So individuals, if I say the big S is normal, which means you're going to have normal hemoglobin, normal red blood cells. I'm going to do this S with the sickle gene. Okay, so if you get a normal gene from one parent and a sickle from the other, that's what we call a heterozygous condition. That means you have sickle cell trait. And normally, you don't really notice any fatigue or tiring or anything like that because at least half of your hemoglobin is still normal. So really where you would see this, maybe someone that's an athlete that does a lot of cardiovascular exercise, they might get winded a little bit quicker than someone else, but pretty much that person's pretty normal. And however, if people get that sickle cell trait from both parents, then they actually have the sickle cell disease. So they're only producing abnormal hemoglobin, and that is a major problem. People with that disorder will have to have regular blood transfusions. So the problem when it is a sickle shape is that it basically can block, it hinders the flow of blood. It can basically block or occlude further traveling. And of course the sickle shape does not hold as much hemoglobin, so not as much oxygen can be carried either. Okay, so a little review. The red blood cell is convex in the middle. That's false. That should be concave. They are leukocytes. Leukocytes are we call white blood cells, so that is false. Contains nitrogen, protein, hemoglobin. No, that's going to be iron. And the red blood cell is not made of multiple cells. It's one cell. So the only thing that's true about that is that red blood cells live about 100 to 120 days. Okay, which statement does not describe a component of the red blood cell life cycle? So we're basically talking about what happens when we break it down and as we recycle the components. Ruptured red blood cells are removed by macrophages. That is true. The organs that are primarily responsible for that are the spleen and the liver. But as I said previously, tissue macrophages anywhere in the body can break down. Hemoglobin is ultimately converted to bilirubin. Remember, first it's biliveridin, which has that green pigment, to bilirubin, which is yellow, and that will be excreted by the liver. That is true. It's going to be a portion of bile. The transporter of iron in the blood is transferrin. That's what's going to carry and allow iron to move from the liver back to the bone marrow so you can resynthesize the hemoglobin molecules. I actually did not get this detailed, so don't worry about that one, but that one is true. Biliveridin is the protein that stores iron. That is incorrect. That is the waste, one of the waste breakdowns of heme. So remember when we break down heme, it goes from biliveridin to bilirubin, and then we also have the iron component, which we have to recycle. Again, we're looking at which of the following statements is true. A reticulocyte, that is an immature red blood cell, and it does not have a nucleus at all, much less one that's multilobed. Multilobed nuclei are only found in white blood cells or leukocytes. Erythropoiesis is the production of red blood cells, not white blood cells, so that is a false statement. Typically, erythropoiesis and red blood cell destruction should be about equal because you want to keep the hematocrit about the same and therefore the blood viscosity the same. So that's not true. And then loss of the nucleus does not cause swelling whatsoever of the erythrocyte. In fact, as we mentioned, loss of the nucleus and other organelles provide space for proteins to come in to help form the hemoglobin. So the true statement, hypoxia, which is a low state of oxygen, increases the secretion of EPO or erythropoietin by the kidneys. Remember, the kidneys are stimulated to make EPO when oxygen levels are low.